Welcome to our exclusive technical tip series. I am Monica Tyagi. I'm the application scientist at Protein Simple Biotechnic. Today, I'm excited to delve into the intricate world of CE-SDS analysis, where we explore the cutting-edge feature of our revolutionary Maurice platform. Let's embark on a journey of discovery together. I'm thrilled to spotlight our comprehensive Maurice family platform. Introducing Maurice C for charge heterogeneity analysis, Maurice S for size analysis, and Maurice, the all in one solution for both charge and size analysis. Moreover, we proudly unveil our latest innovation, Maurice Flex, which not only encompasses the features of Maurice but also offers advanced fractionation capabilities with the charge isoforms for detailed downstream variant analysis. As previously mentioned, today's focus will be on CESDS analysis. Let's look deeper into this topic. For CE-SDS analysis, we provide a CE-SDS Plus cartridge and a turbo cartridge. As the name turbo suggests, it has faster separation time compared with a CE-SDS Plus cartridge, which is about 5 to 8 minutes. It gives you high quality data and it is ideal for high throughput analysis applications. Whereas a CE-SDS Plus cartridge, it has 25 to 35 minutes of separation time. You get superior quality data. It is ideal for analytical development and QC applications. This comes with our ICE Compass for ICE software. It is also compatible with the Chromeleon from Thermo Scientific and Empower from Waters. I would like to highlight here and talk a little bit more about the key features and benefits of using Turbo CE-SDS cartridge. It accelerates the commercialization by significantly reducing the separation time by 80%. You can obtain more critical data by having 1,000 plus samples for each cartridge. It's a familiar workflow similar to your CE-SDS Plus cartridge method, but with a new running buffer. Reduces complexity and base with no top running vial, 2 tanks in the cartridge. One is for the separation tank and the other is the waste tank. Saves on your cost, lower cost per injection. It is compatible with any Maurice after a software upgrade, both Compass and Embed. Single software for multiple applications, which is equal to increased ease of use. Maurice is a fully automated system with an easy-to-follow CE-SDS workflow. Just pop in one of the pre-assembled CE-SDS cartridges, drop in a few reagents, add your sample vials or a 96-well plate and hit start. Maurice performs size-based CE-SDS and protein varying in size from 10 kilodalton to 270 kilodalton. The typical separation time for a reduced sample is just 25 minutes, and for the non-reduced sample, 35 minutes, running up to 48 samples per batch. Maurice conditions the capillary at the beginning of the run with conditioning buffer 1, which is the base, and the conditioning buffer 2, which is the acid, followed by water and the separation matrix. That makes sure that the capillary surface is uniformly charged, so you get optimal resolution and assay reproducibility. Capillaries are then conditioned every 12 injections to maintain surface integrity. The samples are electrokinetically injected into the cartridge capillary. The separation is monitored in real time in seconds as peak passes a single point detector. The peak relative migration times compared to the 10 kilodalton internal standard is reported by the Compass for ICE after the separation is finished. What's interesting is that with Compass software, you have the freedom to seamlessly transition to the recognizable lane view and customize figures labeled to suit your preferences. In the upcoming slides, we will explore the development of CESDS method in detail. Before we proceed further, I would like to highlight some valuable resources offered by Biotechne. That includes Biotechne Academy, the method development and quick reference guides, as well as insightful application notes. The question is, why CE-SDS over traditional SDS-PAGE? The shift toward CE-SDS from traditional SDS-PAGE arise from the challenge faced with reproducibility and quantitation in protein purity analysis. I still remember the laborious process of running gels during my postdoc days. Today, biopharmaceutical companies are increasingly adopting CE-SDS over slab gel due to its numerous advantages. Maurice, an advanced CE-SDS system, stands out for its ability to deliver reproducible, high-resolution data while offering a straightforward and a robust workflow that minimizes hands-on time and reduces user error. Further, it significantly reduces our environmental footprint, being eco-friendly and require lower sample volume compared to traditional SDS page. Let's look into some interesting data from one of our app note comparing SDS page with Maurice CE-SDS for protein purity analysis. When compared with Maurice CE-SDS for protein purity analysis, the SDS-PAGE slab gel approach had significant disadvantages in terms of sample volume used, assay sensitivity, time to result, and the confidence in those results. The figure on the right shows the NIST mat samples under non-reduced condition, which shows a single high molecular weight, and the heat stretch samples show the same major high molecular weight band, but also show additional minor bands at lower molecular weight, which represent stress-free induced fragmentations. The sample analyzed through CE-SDS and Maurice exhibits superior resolution and signal-to-noise ratios compared to SDS-PAGE outcome, attributed to the exceptional resolving power inherent to this technique. For further insight, please scan the QR code provided on the left. Here, I'll discuss how Maurice compares to the SciEx P800 system for the reduced and non-reduced CE-SDS separation for a reference monoclonal antibody, that's the NIST map. Here we compared our results running the reduced NIST map on Maurice with the P800 in-house and with the P800 published data. The figure shows that the Maurice, which is on the left, the P800 in the middle, and the P800 Plus on the right, all baselines were resolved at the 1 mg per ml concentration of the NIST map. All systems were resolved and detected the light chain, non-glycosylated heavy chain, and the heavy chain. Maurice and the P800 Plus both also resolved a 10 kilodalton internal standard, which wasn't included in the P800 batch. This app note will be helpful if you are looking for method transfer from your existing P800 platform to Maurice. What are the key steps for developing a robust CE-SDS method? Using the right concentration of the sample is important to get the optimal results. An optimal reducing procedure for IgG achieves complete breakage of all disulfide bridges and avoid dimer formation. Ideally, your final sample should contain less than 50 millimolar salt to minimize the effect of the buffer or injection efficiency. The pH of your final sample is another critical parameter to consider. These are some of the key considerations for when you're starting to go over the method development. Maurice CE-SDS applications give you everything you'd expect with the capillary gel electrophoresis, like automation and reproducible, high-resolution data, but simplifies things to cut your hands-on time and minimize user error. A simple workflow lets you monitor the degree of heavy chain glycosylation and degree of fragmentation in reduced and non-reduced IgG molecules quickly and easily. And the Compass for ICE software lets you set up the batch and methods parameter, monitor your run in real time, and analyze data for you in a snap. In the figure, you can see the separation of the reduced on the top and the non-reduced at the bottom for the IgG standard with a 10 kilodalton internal standard. Please feel free to scan the QR code for more details on this application. Moving on from standard IgG, let's talk about large biomolecule analysis with Maurice. IgM molecules are gaining significant interest for their promising therapeutic applications. With the pentameric or a hexameric structure, IgM exhibits high avidity, optimizing its interaction with target antigen. This unique configuration not only enhances its binding affinity, but also enables successful binding with antigens even at low concentration. These characteristics make IgM molecule compelling candidate for innovative therapeutic intervention. Their multi-unit structure and potent antigen binding capabilities position them as a valuable asset in the field of biomedicine. Here is the app note if you are interested in analyzing IgM on the Maurice platform, and I'll be talking a little bit more detail about it. Here is the method for IgM analysis, as mentioned in our app note. The IgM samples were diluted with Maurice CE-SDS 1 example buffer that resulted in the final concentration of 1 mg per ml. DTT served as a reducing agent. Subsequently, all the samples were heated at 70 degrees Celsius for 10 minutes, followed by cooling. The treated samples were then loaded onto the Maurice instrument and injected for 8 seconds at 3,500 volt, followed by separation for 14 minutes at 4,200 volt. All resulting data were analyzed utilizing the Compass for Ice software. Let's take a look at the results. A typical IgG molecule, as shown, weighs 150 kilodalton. In contrast, an IgM is known to have a molecular weight of 950 kilodalton. The heavier, well-resolved IgN peak demonstrate that the turbo CESDS method allows for the identification and characterization of the proteins of wide molecular weight range without needing additional method development or modification. The Maurice Turbo CE-SDS cartridge offers high throughput CE-SDS analysis, and large proteins with molecular weights of 900 kilodalton can rapidly be analyzed using the Turbo CE-SDS cartridge. Here is the representative electropherogram of IgM samples under non-reduced and reduced conditions. The IgM subunits are held together by a disulfide bonds, resulting in a 1 big peak as shown under non-reduced condition. Conversely, reduction of the disulfide bond causes the IgM subunit to separate into their individual heavy and light chain, thus altering their electrophoretic mobility. Moving on to fusion proteins and their analysis on Maurice CE-SDS platform. Here are some of the useful resources. Please scan the QR code for the app notes on fusion protein. In this study, etanercept was the molecule of interest. It's a fusion protein developed and distributed by Amgen. This biopharmaceutical compound is specifically designed to address autoimmune disease such as arthritis. As a combination of IgG's Fc region and the extracellular domain of the TNF receptor, it is heavily glycosylated molecule with complex structure, causing additional analytical challenge during its development and manufacturing. For the fusion protein, one of the key differences in the method was increasing the separation time during the method optimization. But if you can look, the rest of the key steps were more or less similar, having the final concentration around 1 mg per ml. Here, beta-mercaptoethanol was used as a reducing agent, then same denaturing by heat, and then separation. The figure here shows 9 representative electropherograms from non-reduced and reduced experiments, respectively. The results were highly reproducible, with the relative standard deviation value being less than 0.6% from the main peaks in both the cases. In the end, I would like to talk about the viral vectors. Here is the useful resource if you're interested in characterizing your viral vectors using our Maurice platform. As scientists are developing adeno-associated viruses and lentivirus-based gene and cell therapies, evaluating the critical quality attributes of your viral vector is routine and required. Instead of using SDS-PAGE for AAV purity and confirming serotypes using PCR or ELISA, you can get data on different attributes of your sample with one platform. In this method, cold acetone was used for the precipitation on the AAV samples. However, there are other methods for precipitation, such as using ethanol or methanol, that you can use. The precipitated AAV samples were dissolved in the SDS plus buffer and denatured by using beta-mercaptoethanol, followed by incubation at 70 degrees Celsius for 10 minutes. The samples were analyzed both on CE-SDS Plus and the turbo cartridge, and all the data was analyzed using the CumbSome software. 4 different AAV serotypes were each analyzed with the Turbo CE-SDS and CE-SDS Plus cartridge. The figure shows stacked profile of 4 serotypes resulting from each cartridge. A distinct profile for each serotype is clearly illustrated through both the methods. The average ratio of viral proteins, VP1, VP2, and VP3 are also comparable between the two methods. For more in details, please have a look at the app note. Here, 5 AAV9 samples with different transgenes were analyzed for their impurities using both CE-SDS methods on the same Maurice instrument. The figure demonstrates how well both the cartridges can detect sample impurities as well as the AAV genome. While there is some expected difference in the resolution between the two, both the methods were sensitive enough to detect minor impurities. A single experiment was enough to provide critical information on AAVs such as capsid protein ratios and impurities levels, showing how the Maurice system is suitable for multi-attribute analysis. The key challenges that needs to be considered and paid attention to is the ionic composition. The salt concentration should be less than 50 millimolar. If it is still high, increase the injection time might help with the signal recovery. And the sample buffer compatibility. Please do check for the compatibility table. And if there is incompatibility, we recommend desalting or buffer exchange. In conclusion, Maurice CE-SDS applications gives you everything you would expect with the capillary electrophoresis, like automation, reproducibility, high-resolution data, simple workflow, real-time monitoring using Compass for ICE software. Thank you for your time and watching the video. Please stay tuned for the next series of tips and tricks.